Is Coby Viner.
Thanks. So today I'm going to be talking about modeling methyl-sensitive transcription factor motifs with an expanded epigenetic alphabet.
I have problems. Let me see. I have to move the. No. You go.
Oh, okay.
Now I will move. Okay.
Thanks. So just as transcription factors have preferences between different unmodified nucleobases, some transcription factors are known to distinguish between unmodified and modified motifs. And these modified motifs often result in different in vivo preferences of these transcription factors. And in particular, it was found by Hugh et al. That for a number of different transcription factors, not only do they prefer methylated sequence, that is, there's a preference for binding motifs containing 5-methylcytosine, often at CpG dinucleotides, but this preference actually results in completely different transcription factor binding motifs relative to the commonly known consensus motifs. And so in addition to 5-methylcytosine, we're also interested in investigating its oxidative products, which are now known, including 5-hydroxymethylcytosine, 5-formylcytosine, and 5-carboxylcytosine. And all of these different modifications can now be sequenced, although it often requires multiple sequencing runs, and this can be costly and difficult to accomplish. So now we have a model of transcription factor binding using the standard DNA alphabet of A, C, G, and T. But we want to additionally model these new epigenetic modifications. And so to do that, we expand this alphabet, and we add new symbols to represent these new modifications: M for 5-methylcytosine, H for 5-hydroxymethylcytosine, F for 5-formylcytosine, and C for 5-carboxylcytosine. And with these different modifications, we have to consider the fact that DNA is double-stranded, and so a guanine represents a guanine base paired to a cytosine. And so if the cytosine is modified, then we want to encode that as well. And so to capture that, we add additional symbols to capture these base pairings to modified nucleobases. So we add 1 to represent a guanine base that is base paired to a 5-methylcytosine, a 2 to represent a guanine base that's base paired to a 5-hydroxymethylcytosine, and so on. And then we add additional symbols to encode certain ambiguities. And so one source of ambiguities might be that you only did conventional bisulfite sequencing. If that's the case, you can't distinguish between 5-methylcytosine and 5-hydroxymethylcytosine. Even if you additionally did oxidative bisulfite sequencing, and you can in principle distinguish these, you might not actually be able to, due to low coverage, for example, at a particular locus. And so to model this, we add ambiguity codes: X to represent M or H, and then on the opposite strand, 7 to represent a 1 or a 2. We also add symbols to represent an unknown modification state, where we have no idea what the modification is, but we don't have sufficient confidence to call it unmodified. And so that's Z or 9. So now we have an unmodified genome sequence that everyone is used to. And what we want to do is we want to modify this genome sequence in order to be able to directly model these modifications. And so to do that, we obtained conventional and oxidative whole genome bisulfite sequencing data from naive mouse T cells from the Ferguson-Smith lab. And using that data, we are able to call different modifications. And the way to do this is to use some sort of software package that can integrate this data. So what we're using is a maximum likelihood approach called MLML, and that's part of the MEthPipe package from the Smith lab at the University of Southern California. And using this, we can integrate this data and get what are like probabilities of modification at different loci. So for example, here we see that this cytosine, we believe with sufficient confidence, is modified to a 5-methylcytosine. This guanine base on the opposite strand has a 5-methylcytosine, whereas here we're not sure if it's a 5-methylcytosine or a 5-hydroxymethylcytosine, but we think it's one of the two. So we can then plot this sequence in the UCSC genome browser and look at its distribution. Another thing we can do is look at the frequency of these bases genome-wide. So for a particular replicate shown here, you can see you have the expected distribution of cytosine and guanine almost 10 to the 8, and you lose roughly an order of magnitude to 5-methylcytosine, which is shown both here on the positive strand and the 1 here on the negative strand, and then yet another of order of magnitude reduction for 5-hydroxymethylcytosine, and then we have a number of bases which have ambiguous modification states. So now using this data, what we want to do is take the sequence information and test particular hypotheses of modifications. So if we have an example unmodified motif as shown here, this is represented by a position weight matrix and visualized here as a sequence logo. We want to test particular modifications. So we can modify the central CPG to be methylated or other modifications. And then we can also modify across all motif fractions across the motif here to be methylated or other modifications. And now what we want to do is take these different hypotheses and ask various questions about this. Where would this motif be expected to bind computationally? Where is it enriched in particular genomes? And so to do this, we use the meme suite. But the meme suite now allows us to do more than that because we can also do de novo motif elucidation. And we've actually now expanded all these tools to be able to work with custom alphabets. And so what that means is we can directly specify M, H, F, or C, or any of these ambiguity codes, and we can do de novo motif analysis and then downstream analyses with the various tools of the meme suite to be able to get at this transcription factor binding with these epigenetic modifications. One of the tools that we make the most use of is Centrimo, and it allows inference of DNA binding from ChIP-seq data. The way it basically works is you scan peak centers with position weight matrices. So here you have ChIP-seq signal, and these represent the peaks where the highlighted region is the central area. So you would scan these and count the number of sequences that are the best match at each position of the central motifs. So here we have example sequence matches. You normalize these counts to compute probabilities, you smooth this, and you plot it. So for this particular example, we have this orange motif, which was found not to be statistically significant, while the blue motif had higher statistical significance than the red motif, but less centrality. And so using this on real data, we can, for example, look at CBP beta, which has a known preference for methylated DNA. And so if we look at this Centrimo result, we can see that all the top hits, the most central and those with highest probability, are all more enriched than the unmodified ones shown here in blue or orange. And so we're able to recapitulate the preference of CBP beta here for methylated DNA. Now at this point what we want to do is we want to use Centrimo to decide upon the modification threshold. If you remember from before, I showed you a discrete genome sequence and said I was calling these of sufficient confidence from various probability-like estimates of 5-methylcytosine and 5-hydroxymethylcytosine data. So to figure out what thresholds we should use, what we do is we try different thresholds and we look at known transcription factors where we know what should occur under methylation. So in particular, ZFP57 and CBP beta are known to prefer 5-methylcytosine, while CMYC is known to prefer unmethylated DNA. So we look for those and use Centrimo to directly compare these different modified versus unmodified cases. We force a constant motif width to do this, and we also normalize Kullback-Leibler divergences in order to account for the fact that these bases are much rarer, and so their background frequencies are lower, and we don't want artificial enrichment of these motifs. So what we then get from this sort of analysis is a plot like this, where I have thresholds from 0.01 to 0.99, and on the y-axis we have what is a ratio of Centrimo p-values of unmodified motifs, in this case all E-box motifs because this is for CMYC, to different modified hypotheses. So here we're testing the hypothesis that CAC1TG is that better or worse in terms of binding than the unmodified version. So because this is log transformed, if it's below 0, that means there's a preference for the unmodified motif. If it's above 0, then there will be a preference for the modified motif. And so if we look at this for CMYC, for 4 different replicates of whole genome bisulfite sequencing data and a particular replicate of CMYC data, we can see that there is a consistent preference, because all these values are below 0, for the unmodified motifs, no matter which modification you test. And then also, as long as you're above a threshold of 0.3, then this preference is quite strong for the unmodified motif. So this recapitulates what's been known for quite a while in the case of CMYC. If we do a similar thing for ZFP57, we can see that almost all of the motifs have a preference, a very strong preference for the methylated or hydroxymethylated motif, and all of them have some preference for those modified motifs because it's always above 0, and so the modified motif is always preferred over the unmodified motif, which is also known. And CBP beta, as I mentioned before, prefers 5-methylcytosine, but it also actually dislikes binding to 5-hydroxymethylcytosine. And we see that here with the ones above 0 being the methylated DNA generally, whereas the ones below 0 are for hydroxymethylated DNA, and that's saying that it does not prefer that, and that's also a known result. And so if we look at all of this information, we can see that transcription factors recognize epigenetic DNA modifications, expanded alphabet motifs can predict differential binding, and Centrimo can estimate expanded alphabet motif binding affinities. So I'd like to thank everyone listed here, and in particular my advisor, Michael Hoffman, our collaborators at the University of Queensland who did the main work on extending the meme suite to a custom alphabet, those at the University of Cambridge and the Sanger Institute who provided whole genome bisulfite sequencing data, and the University of Cambridge again for providing ZFP57 ChIP-seq data. And thank you all for your attention.
Thank you, nice work. I have a question. So you're using the Centrimo tool, which is quite computationally intensive. So I guess that you restrict your set of picks that you consider from the ChIP-seq experiment to like 500 or 600 picks. But in that case, my question would be, would you see a difference in terms of enrichment where you were looking at maybe the top 600 picks, and then when you go a little bit lower to the next picks and so on? Because I think this is something that might be gradual rather than it's really enriched for this methylation in the top one, but it might be different from the other picks, right?
So generally for Centrimo, it is fairly intensive, but it's not nearly as intensive as, for example, meme. And so we are able to run Centrimo across the entire peak set, assuming we already have some position weight matrix that we're testing.
Okay.
And so in terms of what we actually find there, I am restricting in these analyses to the top Centrimo hits with respect to the motif pairs. But generally speaking, I think that as long as you have your position weight matrices, either from hypothesis testing or de novo, you can test those directly.
Okay.
Thanks again. And,
Thanks.
our next speaker